Hi everyone, welcome to researchmd.com. We got a great presentation today. Let me introduce myself. My name is Dr. PRAMIL. I'm a practicing physician in the United States, Program Director, Internal Medicine, Translational Residency, Associate Professor of Medicine in two large medical schools. Today we're going to look at the antibiotic, or we're continuing our antibiotic lecture. Today we're going to talk about beta-lactam antibiotic. We're just going to start. What are they? How does it work? And how does the resistance develop? Very, very important examination topic. Okay, so when you talk about the beta-lactam antibiotic, there's four different classes we need to know. One is penicillin, number two, cephalosporin, number three, carbapenems, and number four, monobactams. Remember the four main classification. Okay, but today we're not going to too much details into the drug, but we're going to teach you what is beta-lactam, how does the resistance develop. That's our purpose, okay? In order to do that, we have to look back at the bacterium. We have to remember we gave a lecture about the membranes a few days ago. Please review that. If you look at the bacterial cell wall, there is something called peptidoglycan. What are peptidoglycan? Okay, you have interlinked N-acetyl glucosamine and then N-acetyl muraminic acid alternating, and then you have pentapeptides linking them together. Okay, pentapeptides are L-alanine, D-glutamate, L-lysine, glycine, and D-alanine. Now, then you have an interlink right here between glycine and cause like a glycine bridge, and all of this kind of make this stronger, making a peptidoglycan bridging kind of together. Now we have to talk about another very, very important protein. What is that? Penicillin-binding protein. Okay, what does penicillin-binding protein do? Okay, it catalyzes the peptide bond between two alanine subunits, and then they form this peptide side chain up here and make this kind of interlink together and making it stronger. It's very important in the bacteria to form this peptidoglycan in the cell wall. Okay, remember that. So now we have to look at our antibiotic. We already said this four classes we talk about: penicillin, cephalosporin, carbapenem, and monobactam. Penicillin we know discovered like by Alexander Fleming in the 1900s, right? What does penicillin and all this have is something called beta-lactam. Okay, so this beta-lactam is actually, you got 1, 2, 3 carbon, and then an oxygen and a nitrogen over here. Okay, this is called a beta-lactam ring. It's a ring, beta-lactam ring. But now, if you look at the beta-lactam ring and the penicillin-binding protein, they got this alanine bridge, kind of pretty much similar right here. Okay, so what it does is like this beta-lactam ring bind to the penicillin-binding protein. Okay, so what happened when it binds to this? This function will not take place. So this wall can break down, no peptidoglycan formation, and everything is going to be affected. Okay, so remember to understand what happened. You got a beta-lactam on the antibiotic, goes and bind with the penicillin-binding protein because of this alanine bridges are similar and they can mimic the penicillin-binding protein that bind to it. Okay, so this process will not take place. Okay, now let's look at how does this produce resistance? That's also important. We're giving a lot of antibiotic now these days, right? Because they develop resistance, they don't work. So we have to combine. We have to make new antibiotic. There are 6 mechanisms the bacteria develop resistance to the beta-lactam antibiotic. Okay, so 6 P's. Let's say 6 P's. We're going to talk about each P first. What is the first P? Right here, penetration. Okay, number 2, P is porins. Okay, then we have number 3, pump. Okay, and then we have number 4 is the penicillinase, and we have number 5 penicillin-binding protein, and the last one is the peptidoglycan. 6 P's, remember, okay? Very, very important because it could be an examination question. So let's start with the number 1, the penetration. So this penetration into the beta-lactam ring penetration into the cell membrane and cell wall is going to be affected because of the mutations, okay? And then number 2, you have porins. These are the channels in the cell membrane and the cell wall, for the bacteria has to transport the nutrients. But you have the mutations, mutated porins. So when you have mutated porins, antibiotics usually, if there's no resistance, they use this porin channels to get into the bacteria. But unfortunately, the resistance develop because of the mutated porins. The antibiotics will not reach the bacteria. Okay? So number 3, let's look at number 3. There's a pump. It's an efflux pump. So whenever this antibiotic gets into the cell, this pump activated efflux, push it out the antibiotic out of the cell. So what happens? The concentration of the bacteria in the cell is very less, so it may not work. Okay, that is the resistance. So we did number 4. What happened? There is this bacteria produce an enzyme called penicillinase. What does it do? They just destroy this beta-lactam ring. They also call it beta-lactamase or penicillinase. So if that, what happens? Then you will destroy beta-lactamase. It's not going to work, right? And the next one, number 5, is penicillin-binding protein itself is going to be affected because of the mutation. If there is no penicillin-binding protein, the beta-lactam cannot combine to it, okay? And then you got like number 6 is the absent peptidoglycan. Remember, we got this peptidoglycan over here, the role of penicillin binding. If there is no peptidoglycan in the cell wall, that's going to be affected with the resistance, and the antibiotic will not work. Okay, so it's very, very important to understand the structure, and just go summarize like one more time right here. So mechanism, we're talking about the beta-lactam, 4 classifications, penicillin, cephalosporin, monobactam, and carbapenem. And think about this peptidoglycan in the cell wall. Okay, so what does it do is, you got this NAG, N-acetyl glucosamine, N-acetyl muramic acid, interlinking, and we got the pentapeptide bridges, and we got glycine bridge in between, and there is something called the penicillin-binding protein. It catalyzes the peptide bond between 2 alanine subunits of the peptidoglycan and make it very stronger. Now we have this new antibiotic. What does it do? They have something called a beta-lactam ring, which mimic the peptide bond. Okay, look at this part right here. This is pretty much similar, this alanine bridging, and over here and here. So what do? They bind with the beta-lactam and the penicillin binding protein, so this reaction will not take, so cell wall can collapse. Remember that. Now, it's very important to look at the resistance, okay? This beta-lactam resistance is a very big examination topic. There's 6 P's we need to know. 1 is penetration, number 2, porins, number 3 is the efflux pump, and number 4 is the penicillinase enzyme and the penicillin binding protein, and number 6 is the absent peptidoglycan. 6 P's, remember that. Thank you so much for watching. We'll be back with another presentation soon. God bless.